be. So also request the participants. I can see around 200 plus participants who have joined us. I request you to please put up your questions in the chat box. And after the talks from our respected speakers today, we'll take up few questions. So please stay tuned. We have a wonderful session ahead.
अब तो
Please
हो
drop your
जाएगा
comments on the chat box. Thank you. Over to you, Smita.
Thank you, Sujit.
अब वो
Now
स्क्रीन
I
बड़ी करके छोड़ दो
request Dr. Aparna ma'am
नहीं ठीक तो
to
है यही तो दिख रहा होगा
deliver
हाँ मुझे
her.
ये दिख रहा होगा
So Dr. Aparna will be speaking on the overview of BIRAC efforts in fostering ecosystem growth in precision biotherapeutics. So
Thank you, Sujit, and a warm welcome to all of you, our experts, Dr. Satin and Dr. Alka Shubha Ghosh, and participants, Dr. Kamakshi and Dr. Vaishali, for this webinar. Thanks for coming to attend this webinar. And myself, Dr. Aparna, I am handling cell and gene therapy and monoclonal antibodies verticals in BioE3 and have been looking into the areas of biopharma. So today I'll tell you a brief overview on the BIRAC efforts and the global and the Indian scenario on the cell and gene therapy. So starting with the global scenario, so it has been a transformative journey in the innovation in R&D from last few years, and especially under the biopharma. CGT has representing the next frontier in the therapeutics innovation, and its biomanufacturing market is witnessing the rapid growth, which is driven by the rising demand of the advanced therapies in the genetic disorders, rare diseases, and cancers. And as you can see, that the market of the biomanufacturing is going to be increased by 2034. So next slide. So when we see that the global cell and gene therapy scenario, that if we see during the last 10 years, there has been an increase in the approvals in the drugs in the cell and gene, and this has been increased especially after the 2021, and gene modified cell therapies has emerged as a dominant modality since 2021. Next one. So this shows that maximum early stage programs are there for the solid tumors, and there is a this that carries a dominant volume. But when we look at the blood cancers, the programs are much more mature. So this shows that there is a lot of excitement for the solid tumors uh, therapies. So next slide. So in this, when we look at the particular therapy area wise, that cell therapies, gene therapies, so we can see that there is a oncology dominance is overwhelming, and it commands the pipeline landscape. And the CNS is the only therapy where people are attempting the gene therapies, maybe because cell-based approaches are not that successful, and this is the only solution for the neurological disorders. Also, the ophthalmology pipeline reflects a substantial concentration of the gene therapy programs. Next slide. So this shows that how it is moving through when people are using the different targets, and CD19 has demonstrated a cross-modality dominance across all. And if you will see that under the gene therapy especially, there is a lot of interest in the orphan and rare diseases, and people are targeting dystrophin, CFTR, and factor 8, 9, which represents the therapies which are being attempted for the rare genetic disorders which have unmet need. Next slide. So now, when we see the Indian scenario under this, the global data shows that there are 7 approved products across the country, and in this, few are supported by BIRAC also. Next, we will see the next slide, please. And this in the Indian scenario, it has been seen that the oncology and the hematological disorders account for 56% of the India's cell and gene therapy pipeline, which shows that this dominates in India portfolio. Next slide. But when we compare this with the global scenario, only 1% of the global cell and gene therapy pipeline is what India is currently, standing for. So there is a lot more has to be done in this area. And as you can see, under the preclinical and discovery, maximum early stage programs are there which needs to be boosted up and efforts needs to be made. Next slide, please. So this shows again that the early stage programs are led into the multiple myeloma, lymphoma, leukemia, and glioblastoma, and hematological tumors and solid tumors, which shows that India has a dominance in the hematological innovation to solid tumors expansion. Next slide. So now this slide actually shows that what is the present momentum in the area, and as you can see, few achievements like there was the first indigenous CAR T therapy which has been developed by ImmunoAct and TMC, and CAR-TME, which is the second CAR T therapy for non-Hodgkin's lymphoma, has been launched, and this is being commercialized, and there are some pipeline studies which are going by the ISTEM where they are developing a gene therapy for uh, retinal cell therapy, sorry, and the gene therapy for hemophilia by CMC. And recently, the CSIR IGIB has developed a CRISPR-Cas9 based gene therapy for sickle cell disease, which they have uh, joined a partnership with the Serum to take it further. Then Miltony with THSTI is giving a CAR-T and gene therapy training curriculum to foster the workforce in this area. And Bharat Biotech has opened a contract research development and manufacturing organization for in this area. So these are the few highlights which has been there in this area in the country. Next slide. So now coming to the BIRAC, like having this all, like giving a brief intro for BIRAC. BIRAC has been established 13 years ago, and the purpose was that DBT was supporting the academic research, but there has to be a central agency which could support the entrepreneurs, medium and large scale startups to develop the high innovation, high risk innovation, affordable products which have an unmet need. And next slide. So that's why the BIREC actually supports the innovative programs under the different thematic verticals, and this area, cell and gene, comes under the biopharmaceuticals and regenerative medicine. Next slide, please. So this is how this program, the projects under the cell and gene is supported under the various programs of BIREC at different stages on which the program is. And next slide. So this slide shows that how in the oncology the BIREC has supported different clinical trial projects, and few are ongoing, and especially they are targeting CD19 CAR T therapy, and one which has been successfully complete is from Immunil, and from IIT Mumbai and INTAS are still ongoing. So when we look at the facilities, then lentiviral vector facilities and cell therapy development manufacturing facilities has been developed, which has been supported by BIRAC. So we will look further. Next slide, please. And this is the first, urological cell therapy product which has been commercialized by Rigro Biosciences, which is an FDA-approved therapy for urethral structure. And this was the one of the successful, cell therapy product which has been supported. And there is also allogenic cell therapy product which was supported for the ophthalmic condition for dry related macular degeneration, and currently this product is under the pipeline in the clinical trials. Next slide. And this is the facilities, lentiviral manufacturing facility which has been supported to INTAS. Next slide. And this is the Immunil Therapeutics for development of cell and gene therapy product under the GMP facility, and they are also carrying out a clinical trial for B-cell lymphoma studies. Next slide. So now, having all this, it was realized that there is an effort to be made for developing the biomanufacturing capabilities in India, and for that, the Bio E3 policy was launched, where there is an aspect of the bioeconomy, increase in the bioeconomy, having an environmental friendly protocols and methodologies, and to generate the employment. So this was next slide. This was actually launched in 2024 by Honorable Prime Minister, and the Honorable State Minister for Science and Technology. Next slide, please. So this policy had a framework for supporting the supporting and intensifying research and innovation, and there was an aspect to boost the domestic biomanufacturing capability to nurture the cohort of highly skilled workforce and having the facilities where the startups can scale up and do a pre-commercialization manufacturing. So these were the few, framework policy under this policy. Next slide. Next slide, please. So very well thought of the six thematic verticals under this policy, which is bio-based chemicals, and Dr. Sharma has already summarized this. And the cell and gene therapy under the precision therapeutics was one of the vertical where discovery stage projects where ideally TRL 3 to 5 support stage projects are being supported, and where there is a after the generation of POC there is a always like stuck, the startups are stuck that how to bridge the gap of taking the POC to early stage commercialization, and for that there is an aspect of supporting the projects under the bridging the gap. And there is a lot of public private partnership which is expected under this program, and there are state connect which is being there, and international collaboration. So next slide. So biomanufacturing hubs and biofoundries are being supported under this program, where there is a aspect of common usage of pilot and pre-commercialization manufacturing facilities, where researchers, startups, and SMEs could utilize this facility. This is accessible to all strategic central agencies, and this will enable and demonstrate the applications and the learnings which can be taken across the other emerging applications, and it will generate a skilled manpower in the area of biomanufacturing. So next slide. And here comes few examples like what has been supported under this. Next slide, please. So these are the biomanufacturing platforms which are being supported, and this is as you can see that there are cell therapy manufacturing hubs which are being supported to attract and TMC and GMP grade facility for gene delivery vector to immunoadaptive cell therapy private limited. So they will have a GMP facility for manufacturing at different scales. Next slide. And then we will have mRNA based precision medicine, where also phase 2 by 3 CAR-T precision medicine for oncology indication is being supported, and biofoundry for the development of high quality precision biotherapeutics to THSPI, where they will have a live biotherapeutic from wild. So this is all is being supported as a part of biomanufacturing program platforms. Next slide. So after having these facilities, there are need of the products in this area, and that was the basis on which the product development calls has been announced under the six sectors, and the projects have been screened, and the support the process is still ongoing to support them. Next slide, please. So, this is the sector where there were no regulatory frameworks, and ICMR with DBT has put down the national guidelines for gene therapy product development for the
अभी एक
Indian
काम करो, एज में खोल के
researchers.
देख लो, यूपीआई
And
थोड़ी देर में भेज
we are
दूंगा मैं
doing
खोल
time
के,
to time the
एज
stakeholders
वाला ब्राउजर
meeting,
होगा
where
एक।
recently we had done a
अरे
stakeholder meeting in cell and gene therapy area.
वो माइक्रोसॉफ्ट का
And
अपना नीचे देखो,
so
ब्लू कलर का
I
एक
think
राउंड राउंड
this
आइकन
is
होगा।
a this will be needed step for this area, where
हाँ,
the
इसमें
research
खोलो, अभी
projects
भेज
are
दूंगा
stuck,
थोड़ी देर में, थोड़ा
and
बिजी है
I
मैं।
think this will be a great help to all the researchers. So I'll stop here, and thank you for listening to me, and we will take the questions. So we will now move to our experts outlook towards this area. So thank you, Smita.
Thank you, Dr. Aparna, for such a comprehensive overview. Next, we would go to Dr. Arkasubra Ghosh, Narayana Netralaya Foundation, Bangalore. for his talk on role of academia in fostering precision therapeutics. Dr. Ghosh?
Thank you so much. I hope my slides are visible.
Yes, they are.
Great. Thank you so much and thanks for having me.
Sir, request you to put on the slideshow mode, please. It's visible to us, but
Uh,
just put on.
it is on not on slide, so
Not yet.
is it now?
No, I think you'll have to share the entire screen for it to go on.
Can we share, Sujit?
Sir, we haven't received actually the presentation, but we had a initial practice session with Dr. Ghosh where it was successfully going. So he was presenting from his end. Sir, if you have a problem, you can send it to us. You can send us in WhatsApp, or you can quickly display that. Yeah.
I think we have lost contact with Dr. Ghosh. I think by the time he rejoins, shall we uh, hear a talk from Mr. Satyen Sangvi on opportunities and challenges in precision biotherapeutics? Sangvi, I'll be sharing the
Yeah,
slides.
yeah. Hi, good afternoon. Can you hear me?
Yes. Yes.
Yes.
Okay,
So,
thank you. Please share. Yeah. Yeah. So, good afternoon and thank you, organizers, panel, and audience. Greetings to Dr. Sharma and Dr. Aparna. Just introduce myself, Satyen Sangvi. I'm the founder and CSO, Chief Scientific Officer of Regrow. Regrow is India's leading cell therapy product development company. We are operational since last 16 years now, and we are the first company to have three cell-based products approved by Indian FDA DCGI, which are in the market. We've treated roughly more than 5,000 patients, helped them to regain back to their active normal lives. We have a team of 80 members, and our GMP R&D center is located in Yonanda in Pune. Next, please. So I just wanted to give you a little bit of a breakdown of cell and gene. Our topic discussion today is about opportunities and challenges. So I'll just go through what Regrow as a company we've been, encountering these challenges, the advantages we've seen, that we've had over a period as a first mover. And through our journey, I'll take the talk. And I think that will be more relatable and more practical. So, as Dr. Aparna already mentioned, cell and gene therapy, you can categorize them mainly into three categories. One is the cell therapy regenerative medicine, which involves autologous and allogenic stem cell based products. Second is the CAR T related products, which are there. We've heard about that. And the third set of products that are there, the category of products, are gene edited cellular products. So if you break down the cell therapy, there are a lot of types of cells in the cell therapy: iPSC, MSC, HSC, NK cells, dendritic cells. These cell therapies are autologous as well as allogenic. They've been initial first cell therapies that have been there starting from the US in 1999. There's a lot of talk
नॉर्मल
about
वाले टी कर देना आज ब्लैक टी कर
different conditions. There are more than 1100 clinical trials that are happening in the US and Europe across phase 2, 3. There are multiple products which are across musculoskeletal, GVHD, burns and wound healing, Crohn's, autoimmune condition, or cardiovascular, urethral, and corneal conditions.
बिस्किट देगा एक दो
So this market is growing. Currently at $5 to $7 billion, and it's going to rise as the time goes, as we are going to see for all the categories of products. CAR-T is important. It serves patients who face imminent death. So these are patients who, if they don't have this treatment, there are very limited options. Um, so there are CD19, BCMA. CAR-T is mainly autologous. We don't have any allogenic products yet, but there's a lot of talk about in vivo CAR-T product development, which is the latest new trend and where investor money is being poured. There are 1700 plus studies going on. There's a lot of countries leading the race. China, Europe, US are leading. And there are a host of products that have already been approved by the US FDA, Europe, China, Japan. And again, the market is $6 to $7 billion, estimated to grow around 30%. There has been a small dip in the CAR-T market because of the unexpected recurrence in the cancer, but investors are very, optimistic about using CAR-T in autoimmune conditions. Gene therapy is obviously high risk, high reward. It is the largest number of products that have been approved. It is serving more than 50% of conditions which are non-oncogenic. Many of them are sickle cell and beta thalassemia, two conditions which affect a lot of Indians. Those products are approved now in Europe and in US. And again, a lot of the big pharma have acquired these products and these companies to get it into market. The annual sales revenue grows between 100 to $200 million of all these products. They cost almost 1 to 2 million per patient. So this is a gist and a global landscape. Next slide, please. So I'm going to break down the presentation into 4 slides. So typically there are a lot of startup and founders here. Hopefully I'm able to relate to them and help them out in this talking about our journey. So there are 4 things to a product. When you have an idea, you start with the product development. You go to developing and having a product. Let's say, what is the type of a product quality? How do you scale that product scalability? And the fourth is the product affordability. Is there a market for it? Are there payers for it? So these are 4 important aspects of my, pillars, or you can say journey points as you'll go through. So the first is novelty. So, it's very important when you make a product, you have novelty. So there are a lot of opportunities and challenges in both. So opportunity is India is $1.4 billion, 1.4 billion people, right? So 140 crore population. So we must focus on India relevant disease targets. Though there's a lot of hype on CAR-T, let me tell you, 95% of oncology cases are solid tumor. Only 5 to 10% are actually blood cancer, hematological malignancies, which is representing only a lakh or 2 lakh of people. Obviously, there should be a product for them as well, but let's not forget the 95% of the people. So again, when you are doing novelty, it's important to have Indian targets. It's important to have your IP in place. Also, try and build, try and have an out licensing or a joint venture possibility versus building your own thing by yourself through all the
उसके
stages, which
लिए
is
से
very
मत दो पहले
difficult.
तो उसको प्रिंटर कनेक्ट
You have again challenges in that sense that there is IP complexity. IP regulations have improved, but there is a long tenure. You have to wait for your patent to be granted. Also, in India, we have a very strong basic research experience, but not so a very large experience in the translational research. We had a brain drain, but now I think it's changing. The trend is changing because of the policies, the growth in the country. It's becoming brain gain now. So I think I'll move that out into an opportunity now. So funding, let's talk about funding. So when you are at your ideation stage, you have a protocol, you have a prototype, sorry. You can leverage BIRAC schemes. There are a lot of these schemes available right from SIBRI, BIPP and all. You have the Bio E3 policy as well. Also, I think strategic pharma help you a lot. You can build relations with them. If not at early stage, you can partner with them at a late stage. You should have a plan of a 10-year funding runway. It's very important to have that. Otherwise, you can stop midway and not really have enough data or value that you've created, which may not give you any returns later on. So again, obviously, as we know, cell and gene therapy related products and capex requires high capital, whether you're setting up a small pilot scale lab or whether it's running some experiments or just having the right type of people in your team to do these studies. Capital is quite high. Um, People, MNCs or Indian companies don't really partner with you in early data partnerships. So if you have your product at preclinical stage, I don't think there's much partnership scope at this stage, though it's changing. And again, we have a very limited biotech VC private equity community, which is now we are seeing some, strategic pharma investing along with the VC and PE recently in a couple of companies that Dr. Aparna mentioned. So that is also slightly changing now. Regulatory wise, when you're developing the product, it's we have CDSCO. They engage pretty early. They help you out. But one must consider global filings as well. That gives you a lot more credit, a lot of more credibility to your product. As far as the regulatory is concerned, we have a evolving framework. We don't have a very established framework. There is an uncertainty on timelines, and definitely the cell and gene therapy pathway is new, and the experts are relatively few. But we are seeing a lot of policy, lot of guidelines coming, which are focusing more on biologics and medical device. And not to forget, we have a very mature clinical trial ecosystem as well. So, founders and startups should really leapfrog into the next generation of products and not really do any sort of a copy paste mechanism as we've been doing in generics and pharma. So, we should try and think of something innovative and really use BIRAC schemes, DBT, funding available and go next level. Next slide, please. So, once you have a product development pathway, you have the funding sorted, you have the, regulatory steps identified, you're really talking about what is the kind of product I want to build. And that's what important is, that there are four aspects to a product that you must have, especially in a cell and gene therapy product. You have to have safety, you have to have efficacy, you have to have durability, and you have to have exclusivity. So these are four pillars to a good product, which can really be a lot of commercial gain for you and your team, whoever's developed it. So safety, you should start thinking about safety from initial time itself, and you should build safety endpoints using leveraging India's clinical experts. We have a lot of doctors in premium government institutes as well as private who are ready to help out in early stage development of planning a product through phase one studies. And I think that's very important to have a risk profile which is going to do well and work out well. Second point is efficacy. How efficient is your product? What is the result? So it has to be a meaningful clinical benefit. There are a lot of companies that plan, I will give 6 months additional life to a patient who's suffering from a cancer. I don't think that's going to sell in India. So you need to think out, is my product adding 5 years to a patient who's terminally ill? Am I giving 2.5 years? So something like that. So there are a lot of products that did not have clinically meaningful benefits and were not approved by reimbursement agencies. So we have to think about that, and not just to the patient, also to the hospital. Are we, is it one-time treatment? That is very important in India. Patients like the concept of one-time treatment. versus doctors' mindset is to have manage and replace. So obviously, this is because of the lack of data. We don't know how good is the product, how long it is going to last. So these are new age products. So they have obviously some sort of limitations, like we don't know if this is going to last for 2 years, 5 years, 10 years. But you have to keep trust in the fact that if you have a product which has shown efficacy in your phase two, phase three studies, it will work well, because these are all on curative platform. Cell and gene therapies are not like your pharmaceutical drugs, where you have to constantly take these medications. So that's one big difference with cell and gene therapy products. uh, Again, coming to last point, exclusivity, very important if you want to scale your product to the next level with any pharma company to make it $1 billion drug. Whether you have your full IP protected, do you have any kind of special designations like orphan drug, breakthrough designations, which US, Europe have, and whether you're the first mover. If you have a first mover, you have a competition which is at least 5 years away. So that's a very important point to get in a new niche area and get in quick, scale up, get your data, and then move ahead. So yeah, this is about quality. We can move next. Coming to scalability, I think this is a big gap. The policy that we heard about right now, it focuses a lot and gives a lot of support to founders and startups to even big established companies, in fact, to build GMP based facilities. See, because manufacturing is the make or break for CGT. If you're not able to make product properly, you don't have the necessary facility, you don't have the vector production lab, or you're not able to scale those kind of cell therapies, it takes, you're not going to have a successful product. You could build out a small scale. There are those suggestions. You can build out a clinical scale lab and then go to a commercial. You could also look at CDMOs at an early stage and talk to them. You can always partner with CCAMP, which is a government undertaking, which has its own non-GMP and GMP kind of facility. They have a whole ecosystem of people who help you out and help you get your product to the next level. Plus funding as well. They connect you with investors. So again, in terms of talent, see, you have the facility, then you need people. In cell and gene therapy, it takes at least 6 to 12 months to train people to get them to that necessary skill level to handle commercial or patient samples. Because once you've taken the sample from the patient, uh, you've received the blood sample or the bone marrow sample. It's your ethical, moral duty, responsibility to get the product out. It's going to be a big disaster if you kind of mix up and you don't have a good QBD system to get the product out in time. And I think we have a big talent pool. India has been known for pharma workforce base, and we are obviously having a lower cost in terms of labor costs, though it's increasing now, but we have the youngest population in the world. So we have to leverage those USPs and try and build, a good team who can do this. We have a team of dedicated 35 people working at our lab, and I think most of the people are working there for an average of 6 to 8 years plus, with the longest person there from day one. So talent retention is not so much of an issue because there are people who really love this kind of a job, who are in the cutting edge technology work. Little bit on automation, because look, at some point when you scale to a very long, very big number, you will need automation to help you out. I think we are at a point where it's going to assist more than actually kind of do everything, because today's systems and you can say the bio incubators, or you have the what the automated incubators, the those are very expensive. They cost you anything between 500,000 to $5 million. The software GMP compliance is a big issue. So I think what you could do is automate certain very complex parts in your manufacturing or your QC and do away with that. And whereas you could focus on the core activity, which is okay to do within manual systems. One other advantage of automation is that it is a closed system, so obviously reduces contamination. But I think we're yet to get there. But definitely, I think even US is struggling with the automation. So there's a lot of work that is being done there. So GMP facility is very important. You need to have the necessary GMP manufacturing team set up. Uh, yeah, next. So I think we heard about product development. You should have novelty. You should get the funding in place. You should know the regulator, the pathway. Then you make the product. What kind of product do you have? Do you have a safe, effective, durable? Do you have an exclusive market for your product? And the third point we heard discussed about is how do you manufacture the product? Do you have a GMP facility? Do you have the people to manufacture that? Are you able to consistently roll out the product? I think the last point is affordability. I think it's a very touchy topic in India. A lot of people believe that India is a very price sensitive market. There are pockets of people everywhere. Cell and gene therapy is an expensive treatment. It is not something that you can compare with a generic drug or a pharma product. So it has to be looked at in a different way. It's an innovative product. All these years, last 40 years, 50 years in India, we've been doing generics. So this is now biologics is basically innovation. So it's going to be expensive. The manufacturing is expensive. The material is expensive.
ऐसे ही बोल दिया
But
करे यार, वो यहीं
it's very
छोड़ना फिर।
pertinent to have a product which is affordable,
जल्दी जो करते
and I think
हैं
India can do that because when you have
यही
innovation,
सब भीड़ लगा के रख देते हैं।
it's one of the most biggest opportunity India has to make affordable cell and gene therapy product. Just give you a small example. We have a product for the cartilage in the knee joint where we harvest the cartilage tissue, grow it in the lab, and that is then reimplanted back by an orthopedic surgeon to young patients who have injured during sports or physical activity. The same product in the US is available from another company, similar product. It costs $50,000, whereas we are selling that in $1,900 in India. So the difference in the cost to patient is very important, and we are able to do that because of the Indian innovation. We have a lot of engineering talent. We have the scale. We have the market ourselves. We don't need to look outside, and we have a lot of Indian talent available as well. So this overall is a good formula for making the product very affordable. This is a case study in itself where Indian companies which have approved products today, uh, it'll be a lot of eyebrows will be raised when people in the US or Europe will know that cell therapies are manufactured here for $1,900 or a CAR T is available for $30,000. So, those are opportunities that are available. We have a couple of government scheme, Ayushman Bharat, obviously. Uh, with that, a lot of patients are now covered. If you can develop a product which maybe costs within that scheme, it could be great. We have MedTech Mitra helping with that. That's an ICMR, Ministry of Pharmaceutical and NITI Aayog collaboration, which helps you get your product from in clinical stages, helps you in clinical trials, randomized clinical trials, and gets you to Ayushman Bharat. So it's a very good initiative again. Um, so people should know about that as well. So yeah, next slide. So just some just some suggestions from our last 16 years learnings. Uh, so regulatory guidelines when we were there were not there, but we approached ICMR, DCGI, CDSCO, and we worked with them. We helped wherever we were asked, and finally the guidelines were available. So, a lot of founders might say, I'm developing something new, I don't need to have guidelines, but you need to have in anything in health, there should be guidelines, and if not, you should work with them to formulate them. Uh, Keeping patients, benefit, which is cure in cell and gene therapy, is the most is at the center of what you should be doing, because if the patient benefits, so does everyone else. We're also proud of saying that our sort of complete supply chain that we have for raw materials self China free, it's self-reliant. So it's very, very important that you develop products, you know what kind of raw materials are going in, where are you depending with all these geopolitics happening. You should have a very self-reliant supply chain. Also, startups should publish high quality research findings. A lot of things that you keep developing over the period of time, first 2 years, 3 years, 5 years, they are worth publishing, they're worth patenting, but usually we don't document that. So it's important. A lot of founders raise a lot of money, but don't invest in the next big thing, don't invest in the pipeline, start investing in other things. It's important to reinvest in the next big project. Also, in cell and gene therapy, I think you have very early signals. When you're building a portfolio of products, some will work, some will not work. So it's all upon your data, your gut, your market feedback that you should kind of stop programs that are not really leading to any results. We've had a couple of failures, but we've been bold enough to let them go early on itself. So that's also important, So we should have understanding of the data. And lastly, connect early with DBT BIRAC. They've been very helpful, funding grants, and we've been pleasantly surprised, supported throughout our journey. And we thank DBT and BIRAC for this opportunity. Thank you.
Thank you, Dr. Satyan. Thank you for your deep insights, and we have a lot of queries I've been seeing in the chat box. So request the grantee, the applicants to please stay tuned, and also request Dr. Ghosh now to please join us, sir. Since we are running short of time, sir, request you to please conclude in 7 to 8 minutes, sir.
Okay. So, are you seeing my screen?
Yes, sir. I'll share it from us. You can just present. Okay. It's visible.
Okay. Let's start then. All right.
Fine.
I won't
Please go ahead, sir.
belabor the point. Precision biotherapeutics. Lots of trials around the world. Lots of FDA-approved products. We need to get a move on. So that's what it is. So there is an imperative for indigenous precision biotherapeutics in India. And coming from the science side of things, there are two main primary drivers of it. The first one is the Indian genome. So what we know from our studies in genetics is that a lot of mutations that are prevalent, or a lot of genes that are usually mutated in a certain clinical phenotype, don't always show up in our Indian patients. So therefore, we do need to have our own line of precision therapeutics here in India. The second, of course, is addressing the affordability gap. As you know, there's a lot of cost associated with CGT products, so we need to have our own IP around the products that we develop so that we can keep these costs low. Then, of course, there are two broader aspects. We want to move on from making generics or Me Too products to original products. And of course, because of our larger population, we do need to have strategic health security in the event of supply chain disruptions because of whatever reasons. So in order for all of that to be brought together, we need to build the clinical research ecosystem. And when we have that is when we will also be able to address our rare disease burden, which is, again, not rare simply because of our population. So what's a scientist to do? So how can an academic center accelerate such a mission? So first of all, academic centers, we are good at the basic mechanisms and basic fundamentals of precision biotherapeutic programs. So genotype-phenotype mapping, biomarker identifications, and figure out which target works best for a certain condition. On a platform invention side of things, there's vector engineering, there's gene editing, enzyme engineering, and then, of course, the entire process of indigenization of these strategies so that they are tailor-made for our population, for our patients. So these are things that traditionally, even in the West, the pharma has not really invested in. These usually come from academic centers. So this first part is a no-brainer of what a scientist needs to do. The second part is where there's very few centers in India doing this sort of thing, is de-risking the translational aspects and production processes. So this is essentially where the academia goes on to publish a paper and then hopes that somebody will read the paper, come to the lab and take up the technology. So I think that's the part where we as scientists need to change our mindset and think of how we can make the product more palatable for the industry or for anybody who is trying to take it to the market. So translational de-risking is ideally grouped into three areas. This is the disease modeling. So we have animal models, we have iPSC-based cell models of disease. So these should be validated, should be available for providing the data quality of the quality required for regulatory approvals, right? So that's an imperative that can only be done in academia primarily. Second is natural history studies. So this is where academic clinical centers, they can come into play and they can develop these registries with continuous follow-up of patients to really chart out how the patient progresses, which patients progress under what conditions, so that when the time comes for a clinical trial, that data and that information can be used by whoever is going to run the trial. And both of these things will therefore lead to regulatory alignment. From the industry standpoint, particularly in India, since there is very little expertise in complex CGTs, we have a lot of expertise now built up in the last 4 or 5 years in CAR T cells, but there's still lack of expertise in, for example, viral vectors or LNPs, etc., which can be better. There is some, but not quite at par with what's there around the world. So we need to really improve. That's what can academia do. Academia can actually develop some of these process science things which are there in many labs. They're sitting in their shelves. Those can be sort of validated in a product-specific process, and then that can be offered to the industry. So this would lead to establishment of GMP hubs and of course workforce training. That's one critical aspect, and that's something that I want to point out to the funding agencies also, that just building GMP facilities, buying a ton of instruments is not the entire game. We really need to have people who can run these instruments, and as somebody who works in the field, I can tell you it's very hard to find people who can run these instruments and do process development. All right, so the second aspect is more on the clinical side, the clinical learning systems. So a scientist should understand what the clinical phenotype is exactly in the Indian patients and not just go by what's reported, so that we really understand, say for example, if you take DMD, is fibrosis more relevant in the Indian population compared to the Western population? So it's an example, just to make a point that things are different because of our genetic makeup. So we need to talk to clinicians, and the clinicians need to talk to the scientists to really guide the early development, so that you de-risk the product as soon as possible. Second is investigator-initiated trials. So investigator-initiated trials are important, and they are available to be done in India. So we can actually take that route, particularly N of 1 or N of 3 kind of trials for the really rare diseases, because that really proves the technology and de-risks the whole process. Finally, this whole process should lead to frugal innovation, and that's where the regulatory bodies also need to understand that how much of data to be asked for what kind of disease. So that's something that regulatory bodies around the world work on, and we should as well. All right, so I'm going to take a quick moment and tell you as an example all of these things that I told you how we are doing it. So in our Netalia Foundation in Grow Lab, we address all three aspects of it. The first one, technology development. So we have these technologies for making the genes work better, right? The same gene, but can produce more durable protein response, more durable mRNA response over time. So that's an important aspect of technology development. We reduce the dosage required for a particular disease by making better delivery vectors, and then disease application. So again, working with the clinicians, figuring out clinicians and patient groups, figuring out how disease works, what stage of disease is it's best to get the intervention in, and finally de-risking the technology by developing the processes to make the final product. So we have a GMP facility that can produce AAV vectors. So again, AAV vectors, these are gene therapies that are approved for it, and you can see all of them are millions of dollars. So obviously that cannot work for India. So in order to reduce these, we have to have innovations, and these innovations for dystrophin particularly, there should be directed towards the dystrophin gene, which is very large in size, so AAV cannot deliver it. So you have to make microdystrophins, which is about 4.2 kb, mini dystrophin about double the size, and then you have exon skipping therapies, which are specific to specific mutations. So in all of these are independent different technologies, but which one should you use? So the key here is that you can fix dystrophin or you can get dystrophin to be expressed using microdystrophin, dual vector mini dystrophins, or exon skipping. But the microdystrophin, because it is smaller in size, has the least force generation. The mini dystrophin, because it's larger, has greater force generation. The exon skipping one has the best force generation. However, by the time you go to a mini dystrophin, the dosage is twice, because it is two vectors that have to get together. So the dosage is much higher. So therefore the cost is higher. In exon skipping, it is hyper-personalized, only for very specific mutations. So therefore it cannot feed the entire DMD population. So you're left with the delivery option of the first one, which is the microdystrophin. So how do you take it forward? So first, of course, you make sure that it expresses the protein. So this is the dystrophin staining, green showing that it expresses the protein. Of course, mini dystrophin and exon skipping also work. But to focus on this, you have to then do lead optimization or de-risking of the construct itself. So then we made many variants of these constructs, which had different domains, and looked at expression in MDX mice, and that's what's in this slide showing all this red. So therefore, we choose the best ones, not just the one that works that can go into a paper, but you actually go in depth into each domain and figure out what would work in the long term. And then comes the platform innovation. So platform innovation is where we are developing a platform for delivery of genes into muscle. And this platform works much better. So it's an AAV9 based platform, works much better than the wild type. So these mutations, they work better. So we've done those sort of experiments to prove that. And then comes the final bridge. So in the lab, we require dosage for the mice, which is easily produced, but for humans, we need to scale up 10,000 times. How do we do that? So we can't just go to somebody and say, make 10,000 times of it. There's no financial reward for it. So we need to somehow bridge that gap. Can we develop a process so that which can be tech transferred to somebody else who has larger facilities? So that's something that we have done. We've developed a GMP facility at our place. And this GMP facility, we developed the upstream and the downstream processes. And these processes, here, there's a lot of process engineering that's done. And this is a lot of grunt work for which we don't really get any big publications or anything. But it does cost a ton of money, a ton of time, and a lot of people. So this is something the academia can do. We just have to choose to do it, choose to move our focus from papers and more towards developing of a product. And that can really help the country move forward, I think. All right. So to foster the academic CGT ecosystem, of course, there's a role for the funding agencies. Dr. Aparna has already spoken about it. there's a very important role of the regulatory agencies. So we really have to make the regulatory sandbox idea happen. We do really need to work on the single window clearance, and the advisory and monitoring committees need to have some harmonization across different kinds of studies if the platform is the same. So to do all of this, strategic private PPP models, public-private partnerships need to happen. And I think the regulatory bodies are working on handholding models, and I really hope those succeed. So as a final note, what would I want as a scientist in the field? First of all, an agile support framework. What I mean by that is that the grant funding mechanism should move as smoothly as possible. There should be no delay. There should be large block grants which sort of take care of a lot of flexible work. So if I am unable to use a certain amount of material or funding for, let's say, consumables, I should be allowed to get an instrument for it, and so on, or vice versa. If I wish to bring in, say, for example, an expert from outside or an expert from industry, there should be some sort of a method to be able to do that, so that the process and the progress happens better. So dual appointment frameworks, fast track recruitment. So this would really give us the impetus to move fast in a very structured manner. So with that, I'll thank all my funding and my team for the gene therapy work. Thank you.
Thank you. Thank you so much, sir. You have quickly summarized, I think, most of the participants listening to us, they have, I think, most of the queries you have solved, I think, in a nutshell. So just to open up, we'll take very quick few queries. Due to time constraint, we have to close this webinar soon, but we'll just take 3 queries from the participants and request the participants to ask us the remaining queries in the mail, which I've already provided in the chat box. So the first question is to Sharma sir, Dr. Sharma. So thank you, sir. They have all for your opening remarks and a brief idea of the entire biomanufacturing concept, which has, I think, enlightened the participants who are with us. So, sir, the question to you is from Dr. Raza from Lucknow. From IIT Lucknow. Okay, so he's asking, where can we get an in-depth idea of the current scenario of biomanufacturing, especially cell and gene therapy in India? If there is any link, please suggest. Question to Sharma sir.
See, for bioetry and biomanufacturing, we have the bioetry policy document. Probably you can mention that in the chat box, that link you can give. For CGT, actually, Dr. Aparna has covered in her presentation. I think this recording is available in YouTube. That can be accessed.
Yes.
If they have any further questions, if they can write to us.
Sure. Thank you. And also, like Sharma sir has mentioned about the link, I have already put him in the chat box. Please go through the guidelines, RFP implementation plan for biomanufacturing, and also specific to cell and gene therapy, which was provided last year. So this will get an idea to the entire startup ecosystem what exactly DBT and BIRAC is looking for in this area. And also, like sir said, Dr. Aparna's presentation and also our speaker's presentation in a form of recording is uploaded, which you can access in Bioetry websites, also in BIRAC website as well. I think Dr. Smita has a question.
Yes, thank you, Dr. Sujit. The next question is for Mr. Satyen. Dr. Matthew from IIT Hyderabad is asking, India still depends heavily on imported vectors, cytokines, and GMP grade reagents. What strategies can help develop domestic supply chains?
So good question. So one of the important thing is to be self-reliant, and I think that itself is a market in itself. And I think we've seen that in this new policy that Dr. Sharma and Dr. Aparna mentioned, that there is a lot of funding available to grow the own media. So for example, you will not find many cell culture media producers in India. High media is there, but then apart from that, what? So there is a lot of scope, even plasticware, to grow these tissue culture flasks or the next level cell incubators. All of that is not available. So that itself is a big market. So if there are medical device, biomedical engineers, that itself is a big product development you should do. So yeah. So, I mean, but I don't have a direct answer for this question, but you have to, for us at least, we have looked at non-China sources. We look at New Zealand, we look at Australia, we look at US. These are the places we have to rely on, where at least the current geopolitics is, whichever way it is tilting. So that is one thing that founders will have to. I mean, there's very big opportunity.
Thank you, Dr. Satyan. The next is for Dr. Ghosh. Sir, this is from Dr. Sanat. Sir, the question is like, what are the major bottlenecks while translating cell and gene therapy from research lab to GMP manufacturing, and how do we address batch-to-batch variability, especially in autologous therapies?
Yeah, that's an open question worldwide. So the primary hurdles when you move from labs scale to GMP scale is in the term itself, the scale. So usually in the lab you're doing it in a few small plates, and then you move on to bioreactors. So everything changes. The behavior of the cells, the behavior of the reagents. Let's say if you're transfecting something, the ratios of transfection, they change. The dynamics of cell death change. The kinetics of the ATMP production change. So this is, you have to use DOE or design of experiments to actually optimize those things. The second thing about the hurdles essentially is the higher you go in scale, the lesser number of vendors are available for it. And that's something that the previous speaker also spoke of. So you'll really have to figure out your supply chain at that point. So that's another aspect of it. The final aspect of it is QA/QC. So usually that's the critical thing about cell and gene therapies. Each of these products have their own potency and quality check assays, and you have to develop it specific to that product. So there is no standardized product that you can just buy off the shelf for doing these sorts of batch release assays, etc. So that is something that requires its own development cycle.
Thank you, sir. Thank you for summarizing all the aspects. So I see a lot of questions on patents. Request you all to please, I have written in the chat box where you can mail us your queries. For patents, we have a dedicated patent department in BIRAC. We'll be happy to take up this. There's a BIRAC Path program where this will be taken. So request you to, due to shortage of time, we have to close the session. And I request you to please write us in the feedback from after the webinar. You'll be getting a feedback from the registered mail address. Request you to please fill up that and send us so that we can address to your any other queries, if any. And I'd really like to thank everybody, all the participants who have been patiently listening, and like to inform our experts that we have around 290 plus people have joined for this 1 hour session. So thank you to all the participants. And also like to request since we are regularly conducting these biomanufacturing webinars to create awareness among these startups or industries or academia. The next webinar on precision biotherapeutics, that is under, and we'll be taking up the mRNA therapeutics actually. So that will be taken up in the tentatively on the first week of March. Please stay tuned on the BIRAC website and also on the BIRAC social media platforms. So request the entire ecosystem to please be with us and listen and keep checking the regular updates in BIRAC website also. So that's all from my end. Over to you, Smita.
I would formally like to thank our esteemed speakers, Dr. Ghosh and Mr. Sangvi, and also Dr. Sharma and Dr. Vaishali and Dr. Kamakshi from DBT, and all our participants for being patient and interactive throughout the session. Thank you all. And with this, I would like to close today's session. Have a good